Melanosomes or bacteria or melanosomes. You know, in the morning they had already defined melanosomes, but I'm just going to run the definition once more. These are tiny rod-shaped or spherical-shaped structures which impart color to a variety of vertebrate integuments. They are also important contributors to the avian color gamut. But whenever it comes to exceptionally preserved fossil soft tissue, the identity of these microstructures is taken with a pinch of salt. Critics prefer to stick to the earlier notion of whether these are bacterial mats growing on fossilized soft tissues and preserving them, or the other clan prescribes complex batteries of tests to examine their identity. How far do these structures actually resemble bacteria? Now, bacteria come in all shapes and sizes, not just rods and spherical ones. And most commonly with these soft tissue preserved in fossils, we see rod-shaped and spherical-shaped structures. But why not the others? Sometimes we even see these focused into sharp zones. Now, would you expect a bacterial mat to show you such sharp zonation? I'm not sure. Now, what is our key objectives of the study? We actually studied the decay of pigmented and unpigmented chicken feathers, and we used in bacterial inoculum from natural settings which mimicked the ancient Lagerstätten settings, and then we grew bacteria. Now we observed the bacterial morphological repertoire. That means we observed how many different types of bacteria were colonizing this feather, and then we recorded the size and shape parameters in terms of long axis, short axis, and then we calculated the aspect ratio, which is a standard for melanosomes, I'm led to believe. Now all this has been done to analyze the bacterial morphology in context of some fossil avian melanosomes. Now what did we do? So we took pigmented and unpigmented chicken feathers. We ran them through two different test methods. First, we put a set of them in isolated ointment jars with inoculum from mudflats of Aust Cliffs and Portishead. Thank you, Dave. And then we subjected the second set to a still saline water tank with cyanobacterial blooms and a mixed assemblage of microflora and fauna. We harvested feathers periodically after a week, 2 weeks, and 4 weeks, and then we processed them so that we could visualize them under a scanning electron microscope. We measured the bacterial growths, and we measured the bacterial growths in terms of long axis, short axis, aspect ratios using imaging. Now the results. Now this shows you a long axis, the short axis, and the aspect ratio profile. In red, we have the fossil avian melanosomes, and at first glance you can see that the morphology is quite conserved. None of these have as large ranges in bacterial samples. The blue one is from literature, Ichida et al. 2002, figure 6, and these are the green is from the ointment jar, and blue are from the tanks. I apologize, the sizes may not be as big as I hoped it would be, but one thing you can see that the ranges are quite large for bacteria, and secondly, there's a positive skew of bacterial means away from the median in all of these cases. So what we tried next was, I apologize again, it's not as visible. We tried using MANOVA to actually integrate all these factors into our analysis, and what we found was that most of the variation was being explained by long axis followed by the aspect ratio and the short axis. We only received overlaps with high aspect ratio bacteria when we compared them with melanosomes, But these melanosomes had a predicted color of gray and some imparted structural coloration. Now we've always had a problem with these melanosomes which imparted structural coloration, gray, and maybe penguins. They are typically high aspect ratio. But we never received any results which indicated that black and iridescent color, which was predicted for these two samples at the bottom, had any overlaps with our bacterial samples. So what do we observe from taphonomy? So we saw that there was non-selective growth in terms of pigmented and unpigmented feathers. And this was, mind you, this was a surface growth. I did not see bacteria penetrating into the interior. And secondly, the bacterial growth was exclusively along the calamus and the rachis. Now these areas are frequently absent from the genome material. From the morphology, we can explain the skew that we see in bacteria in terms of binary fission. Because what happens is a cell becomes larger, a cell increases in length, then a constriction appears widthwise, following which the parent cell divides into two daughter cells which repeat the process, which is in fact what I believe is causing the mean values to move towards larger values in the distribution. Secondly, we also saw bacterial hyphae on the surface, something that is not characteristic of melanosomes. So, thank you.